Okay, very good morning, my dear friends. Welcome to today's session. Today, our eminent resource person, Dr. A. Madhuri Madam Garu, Assistant Professor, Department of Microbiology, Government Degree College for Women, Autonomous, Begumpet, Hyderabad. Madam is going to explain today one of the important area in genetics, that is backcross and test cross and our other related information. So my dear friends, utilize this wonderful opportunity. Madam, please start your session. Madam, if possible, please enlarge this slide. Please click on slideshow. Yes, madam, it is clearly visible.
Good morning, sir. Thank you.
Madam, your voice is very low. Please loudly.
Yeah. Good morning,
Okay.
dear students. Today, actually, I wanted to take a gene mapping topic. Sir, Tirupati, sir.
Madam, please proceed.
You might have seen previous question papers. Will there be any questions on mapping, gene mapping, knowing the distance between the genes?
Okay.
అంత డెప్త్ లో అడుగుతున్నారా లేదా అని ఒక క్వశ్చన్.
No problem, madam. That is necessary. Any other competitive exams also.
Because in the syllabus it is given 2-point test cross and 3-point.
Okay, after class, definitely I will send you, madam. Please, you proceed.
Okay. So generally these concepts will be used to know the distance between genes. Okay, this is very interesting. But before that, you have to know some basic concepts like linkage and crossing over. Okay, so here is independent assortment always the case? Independent assortment means the genes moving independently to the offsprings without any recombination or without any linkage or without any crossing. Okay, so whatever Mendel gave the ratio, dihybrid cross ratio 9:3:3:1. If that is the ratio, then there is independent assortment, like genes are moving independently without any crossing over, or they are moving to another generation, that is to the offsprings. But as I told you in the last class, Mendel, when he has done experiments and whatever laws he has proposed, his results were according to, or his laws were according to his results. There is a coincidence, right? But after Mendel, whoever scientists have done experiments, they could not get the exact ratio. They could not get exact ratio 9:3:1. So what could be the reason? What could be the reason? Or you can say there is no independent assortment. Always the genes may not be transferring independently to the next generation. So what is the reason? Reason is linkage, or genes are linked. Means what? Just now we have discussed, if the genes are present on different chromosomes, yes, they are not linked, or we can get this ratio 9:3:3:1 as they are showing independent assortment. Okay, if the genes are located very close on the same chromosome, they are showing linkage. Okay, when they are present very close to each other on the same chromosome, we call them as completely linked, or they are showing complete linkage. They may be so close to each other that cannot be separated by recombination. Okay, if you imagine a chromosome where two genes are present very close to each other, can you imagine any crossing over between them? If there is a distance, there can be a crossing over. When there is very less distance, right, there cannot be crossing over, or in other words, they will be going together. That is linkage. Okay, here you see my genes located far apart on the same chromosome typically show incomplete or partial recombination. Here we are talking about chromosomes. See, this is one chromosome, this is one chromosome. These two are homologous chromosomes. What do you mean by homologous chromosomes? Anybody? This session should be interactive, ma. Whoever can answer, please answer fast.
Same recessive and dominant.
What do you mean by homologous chromosomes? It is a pair of chromosomes which are having same sequence of genes. Understood? Humans have how many pairs of chromosomes? Fast.
46.
How many pairs I asked?
23 pairs.
23.
23 pairs.
Very nice. So when we are saying 23 pairs, why we are calling them as pairs? Because two chromosomes are same to each other. Two chromosomes are similar to each other. Okay, that pair we are calling them as homologous chromosomes. Is this clear? Two chromosomes, they are carrying same set of genes. Is that clear? Yes, ma'am. Please be interactive. Two set of chromosomes are carrying same set of genes. This chromosome is carrying same set of genes. This is carrying A, B, C genes. This is carrying D, E, F genes. Both these are carrying same genes. Okay, and these are called alleles. This is called allele, or the position occupied by allele is called as locus, and one set of gene is called as allele. Okay, here these are two different chromosomes, two different set of homologous chromosomes. Okay, so here this is one gene and these are the other genes. Can you see distance between A and B? A and B madhyalo distance unda? Fast. You can unmute
Yes, ma'am.
your mic. Yes. B and C madhyalo, is there more distance or less distance?
Less distance.
Very less distance. Is there a possibility of crossing over between these two genes? Unda leda? Yes or no? Possibility of crossing over
No, ma'am.
between A and B. Between A and B, there is more distance. So there can be a crossing over between these two genes. But between B and C, there is less distance. So there is no possibility of crossing over. ఇది అర్థమైందమ్మా? ఇక్కడ క్రాసింగ్ ఓవర్ అయ్యే పాసిబిలిటీ ఉంటుంది because there is a distance. Here there is no possibility of crossing over because there is no distance. Am I clear?
Yes, ma'am.
ఓకే, సో ఇక్కడ మనం లింకేజ్ గురించి మాట్లాడుతున్నాం. లింకేజ్ అంటే ఏంటి? ఎక్కడైతే క్రాసింగ్ ఓవర్ కాదో దట్ ఈస్ కాల్డ్ యాజ్ లింకేజ్. ఓకే, ఇక్కడ క్రాసింగ్ ఓవర్ అవుతుందా? కాదు. సో దీస్ టూ ఆర్ జీన్స్ ఆర్ సెడ్ టు బి లింక్డ్. These two genes are said to be, yes, linked. Why we are calling them as linked? Because there is no chance of crossing over. These two genes will be going together to the next generation. ఎవరైనా దీన్ని విడదీయగలుగుతారా? Can anybody separate these genes? No. So these are going together to the next generation. That is why these genes are called as linked genes. On these chromosomes, what are the linked genes? This set, what are the linked genes, ma? Fast.
E and F.
E and F. Very nice. My lecture speed will depend on your answering speed. Okay. So these and one more concept is all the genes present on one chromosome is called as linkage group, and here we are calling A, B, C as linked genes and A and D as unlinked genes. As they are all present on the different chromosome, there is no always they will be going separately. Whereas talking about A, B, C, just now what we discussed is, if there is a distance, there is a possibility of crossing over, but crossing over possibility, it is not like always, it is not 100%. It does not guarantee that every time crossing over should happen. So if there is no crossing over, definitely A, B, C will go together, then in that case, these are linked genes. Okay. One sentence you remember, all the genes present on the same chromosome, they are called as linkage group, or they are considered to be linked genes. Next. So genetic linkage, when genes are going together, it is called as linkage, and they are inherited together during meiosis, and the position is called as locus or loci, and if they are present on the same chromosome, there is a possibility of crossing over. And okay, last class we have seen one bit, Bateson and Punnett. They have worked on linkage concept, and the plant they have worked is Lathyrus odoratus, and they have done experiment. They have done dihybrid cross, and they could not get this 9:3:3:1 ratio, which is shown by Mendel. This is the result. This purple and long pollen, two characters taken are flower color and pollen size, okay, pollen shape. So these two are the parental characters. They got in F1, all the offsprings are showing the same phenotype. And in F2 generation, we should get 9:3:1 ratio, but this ratio is not following that pattern. Okay, the results of this F2 offsprings are not following the ratio of 9:3:1. So what is the reason? The reason is the genes are linked, or genes are showing linkage. Types of linkage, they can be complete linkage or incomplete linkage. Okay, here you see complete linkage and incomplete linkage. Here we have taken different characters, that is black body and purple eyes, gray body and red eyes. Okay. Complete versus incomplete linkage. If there is a complete linkage, means what? Complete linkage means genes are going together. Genes are going together. In other words, there is no crossing over. Am I clear? This is a parent. This is one parent. This is one parent. These two are gametes. Okay. So these two are similar. We are taking one gamete. Small r, small g. Here we'll get two gametes. One is this and the other one is this. And we are saying this is complete linkage. Okay. So no crossing over. So what will be the combination of the offsprings? This gamete and this gamete. One pair. And this gamete come here and this gamete will come here into the another offspring. So what will be the phenotype of the offsprings in this cross? Okay, we are getting capital R, small r, and capital G, small g. Capital G, small g, capital R, small r. So the phenotype will be gray body with red eyes. Gray body with red eyes. 50%. And another 50%, small g, small g, small r, small r. That is black body, purple eyes. Over and linkage are basic. So you will get questions from this. Okay. Linkage are there? Yes. Complete linkage and incomplete linkage. In this complete linkage, we have taken these two parental combinations see, this is one parent, same as this, and second parent, same as this. Complete between A and B. Distance between A and B is possible of crossing is what happens as we first crossing over at this point. Crossing over at this and sir, am I audible? Hello?
No, ma'am.
వినపడుతుందమ్మా? ఎస్ గర్ల్స్, ఎక్కడి వరకు విన్నారు?
Excuse me, madam, your voice is not clearly audible, madam. Please let you tell once again, madam. Our students is asking.
Okay. Okay, sir. Right. Complete. Again, shall I repeat the entire slide, ma? Complete versus incomplete linkage. Sir, am I audible?
Yes, madam, it's clearly audible. Please proceed.
Okay, fine. So this is complete linkage. This is incomplete linkage. Okay, incomplete linkage. When you are saying genes are completely linked, means there is no possibility of crossing over. This is simple cross. Okay. Here you are saying incomplete linkage means there is a possibility of crossing over. Incomplete linkage means genes are present on the same chromosome, but they are different at a different place, or there is a distance between these two genes. So there is a possibility of crossing over. Okay. So coming to this parental combination, that is capital R, capital G, small g. Okay. If there is crossing over, where will the crossing over occur? If this is a chromosome and this is a chromosome, there is a possibility of crossing over between two genes. So here there will be a crossing over. Okay. So capital R here at the top and small g at the base. Capital R, small g. This segment will go to this chromosome and this segment will go to this chromosome. Okay. So capital R, small g, small r, capital G. These are the products of crossing over. Am I clear? ఇక్కడ వరకు అర్థమైందమ్మా? Please interact.
Yes, ma'am.
Okay. Next. So we got the combinations or the products of crossing over here, and these are the parental combinations. This is the recessive parent. This is the hybrid parent. Okay. And these are the results of crossing over. Resulted gametes of crossing over here. So what are the different combinations? This is the male parent, this is the female parent, and these are the gametes of female parent of the recombination. So what are the different possibilities? Let us see. Okay, this gamete can have a partner of this one or this one. Okay, so r, small g, along with this another combination can be capital R, capital G. Okay, so we if we write the genotype, capital G, small g, capital G, small r, capital R, small. This genotype ma gray and red eyes, gray body and red eyes. These are 47%. How many offspring? Okay, going to the next combination, this gamete and female gamete. Now in the first case we have taken this gamete. Now we go for this, right? So they have fused and fertilization has occurred and we got an offspring. So what is the genotype of this offspring? small g, smaller, smaller. What is the recessive character here for gray body? Black body is the recessive character. And smaller, smaller. Red eyes recessive character is purple eyes. So the offspring is having black body and purple eyes. Is this clear till now? Yes?
Yes, ma'am.
Okay. Going to the next one. Now again, male parent should be there. Okay. So male gamete has come. Here we are having two recombinant. Okay. So one gamete has come here. One male gamete and one female recombined gamete. Okay. So what is the genotype here? Small g, capital R, smaller. Small g, capital R, smaller. Means small g, what is the recessive character, ma? Black body. What is capital R smaller? Red eyes, because capital R is dominant over smaller. So red eyes, black body, red eyes. And what is another combination? Male gamete, that is small g, and recombined gamete, this is smaller capital G. So what will be the genotype? Capital G, smaller, smaller. So capital G, small g will give gray body, and smaller will give purple eyes. Is this clear? Pillalu.
Yes, ma'am.
Please write. Okay. So now you see the percentage, 47% of this combination, 47% of this combination, 3%, 3%. Is this cross following 9:3:3:1 ratio? Follow అవుతుందమ్మా? Yes or no? 9:3:3:1 ratio follow అవుతుందా లేదా? Bhavna. Yes or no? Just tell me yes or no.
No, ma'am.
No, it is not following. Okay. Why it is not following? Because there is a linkage. Okay. And another point here, these are showing higher percentage and this is showing lower percentage. What is percentage here? What it is showing? It is the number of offsprings showing this character. Number of offsprings this showing this character. If there are 100 offsprings, okay, 47 are showing this phenotype, or 47 are showing this phenotype, 3%, this phenotype. Okay, and one important point, whenever you will get to the number of offsprings, in that whatever is a majority of the number, that will be the parental combination. That will be the parental combination. So these are parental combinations. Just check capital G, small g, capital G, small g, capital R, smaller. This is one parental combination. That is why you got higher percentage. And small g, smaller. This is another parent. Okay. So these are parental combinations. That is why the number of offsprings are more. And these are non-parental combinations or recombinant combinations. That is why the offspring number is less. Clear, ma? Next. So what does linkage show? Linkage will change the ratio. Okay. It is not same as the ratio given by the Mendel. So by linkage, the parental combinations, in just now we have seen parental combinations will show higher number. Linker genes are not inherited together every time. Chromosomes exchange homologous genes during meiosis. Just now we have discussed in the first slide, whatever genes are present on a single chromosome, they are called as linked genes. But sometimes there is a possibility of crossing over. Here we have taken purple chromosome, green chromosome. Okay, but see this part has become green here and this part has become purple here. So what is the reason? There was a crossing over. That is why these two segments have been exchanged. There is a crossing over, right? Now, and always the crossing over will happen between non-sister chromatids. These are sister chromatids. See, these are sister chromatids. Crossing over will happen between or occur between two non-sister chromatids of a homologous chromosome. This is also an important point you have to remember. So linked genes, why they are not going together? Single chromosome, why it is not going as it is to the offspring? Because there is a possibility of crossing over in prophase 1. This is also important point, ma'am. In somatic cells, what cross will occur? Anybody, meiosis or mitosis? Somatic cells or normal cells? What type of
Mitosis.
mitosis? In germinal cells? Yes. In
Meiosis.
reproductive cell, meiosis. Very nice. Okay. In normal cells, mitosis will occur, and in germinal cells or reproductive cells, there will be mitosis. Okay. Sorry, meiosis. Okay. Due to meiosis only, the chromosomal number will be become half in the gametes. Gametes lo half number of chromosomes untai kada. In the last class we have seen. If a human cell is having 46 chromosome, egg or sperm will have how many chromosomes?
23 chromosomes.
23 chromosomes. Okay. So why that number is becoming half there? Due to meiosis type of cell division. Okay. So what is meiosis? Meiosis will have two divisions, two parts, meiosis 1 and meiosis 2. Okay, meiosis 1 and meiosis 2. In the meiosis 1, there will be different cell divisions, stages. In that, in prophase 1, you have to remember, in prophase, there will be a crossing over. In prophase 1, there will be a crossing over in meiosis. Here you see DNA replication. You understand, no? Cells has to divide. So there should be replication. So there is a replication. There is mitosis. In mitosis, if these are the actual chromosomes, the chromosomes have replicated, and during mitosis, chromosomes have been separated and two cells are formed. These are identical to the normal parental cell, and there is no crossing over. These are identical. Okay, coming to the meiosis, meiosis 2. You have to understand always meiosis 2 is equal to mitosis. Meiosis 2 is equal to mitosis. Whatever recombination, whatever crossing over is occurring, that is always occurring in meiosis 1. That is always occurring in meiosis 1. Okay. So in meiosis 1, what is happening? There is a crossing over. That is why there is exchange of these chromosomal segments. This is meiosis 1. Here crossing over is occurring. In which stage? Just now we have seen. In meiosis 1, which stage? Crossing over is occurring? Prophase 1. Very nice. So these crossing overs are causing genetic variation. If you take your own example, are you similar to your father completely? Looks? No. Are you completely similar to mother? No. There is some variation, little variation. That variation is due to this crossing over. Variation is due to crossing over. Okay, clear? How much time is left? Okay, we are having some time. When crossing over occurs, the genes that were previously linked becomes unlinked, creating 4 different types of chromosomes. Here you see, we are having 4 type of combinations. This is parental, and this is recombinant type, and this is recombinant type, because there is a crossing over here. Understood? So how many gametes we got? 4 types of gametes we got. Clear, ma? But the parental combination is having higher percentage, and recombinant combinations is having lesser percentage. Now, a random exchange of DNA between 2 non-sister chromatids. Here, a question will be asked. Crossing over is a genetic exchange between sister chromatids, non-sister chromatids. It occurs in between non-sister chromatids of homologous pair of chromosomes. Homologous chromosomes. Why we are calling homologous chromosomes? We have discussed. Yes? Between homologous chromosomes, we will see this crossing over and that is taking place between non-sister chromatids of a homologous pair. Okay, so there is a crossing over. Now these segments are exchanged, and these are parental combinations, and these are recombined. Sorry, these two are recombinants, and this is parental combination. Now, this is also the same thing. There is a crossing over. Here you can see capital A, capital B, capital C. After crossing over, what has happened? Capital A, capital B. Here, small c. Small a, small b, small c in parent. In offspring, small a, small b, and capital C. So this is the product of crossing over. So these are all the factors which are affecting the crossing over. Just you can go to, and these are all not required. Single crossing over. There can be a single crossing over, double crossing over, multiple crossing overs in a chromosome. Okay, this is the double crossing over, and this is the triple. How many points that crossing over is occurring? That is the number. Okay, if 3 points there is a crossing over, triple crossing over. If 2 points there is a crossing over, double crossing over, like this. Okay, and can be multiple also. So what is the difference you understood for linkage and crossing over? Anybody? Linkage means always the genes are staying together. They tend to stay together. Crossing over means it is a process of recombination where the genetic segments can be exchanged. Okay, so this is about the linkage and crossing over. Sir, how much time is left?
Madam, only 5 minutes left, madam. If possible, please we join once again. Only 5 minutes are left.
So we will break the session. At least 20 minutes I can take.
Madam, join once again.
Yeah. Now
Okay,
itself.
I will stop here. I will end this session, and all of you please join through same link, madam.